Welcome to today's Refresher Lecture. I'm Dr. Leslie Blankenship-Williams, and today's Refresher Lecture is going to cover the second part of aerobic respiration. In a previous Refresher Lecture, we covered how glycolysis and the citric acid cycle played into this big complex process known as aerobic respiration. We'll do a quick review of where we stood at the end of those two processes, and then we'll finish out the process of aerobic respiration. In order to do that, we need to review the mitochondria cellular anatomy and see how that plays into the final step, which is called oxidative phosphorylation. So let's start with where we were. The overarching chemical pathway known as aerobic respiration starts with glucose, adds 6 oxygen molecules, and through many steps produces 6 carbon dioxides, 6 waters, and 38 ATP. Thus far, what we have done in the previous 2 steps of glycolysis and the citric acid cycle is we've taken glucose and we've broken it down into carbon dioxide. But we haven't gotten the 38 ATP that we were promised. Instead, we ended with 4 ATP plus a whole handful of these coenzymes called NADH and FADH2. Nor have we really seen oxygen come into play, or water. So hold those thoughts and understand they're going to come into play in the very last step. Okay, a quick review of basically those 2 previous steps, glycolysis and the citric acid cycle. Recall that in glycolysis we started with glucose and we broke it down into 2 pyruvates. When we did that, we got 2 ATP and 2 NADHs. From there, we took those pyruvates and basically did a prep step. We shed off one of the carbon dioxides, as shown here, and since carbon dioxide is a waste, it gets blown off into the cell and eventually comes out of your lungs during breathing. And what did we get in that step? We got 2 NADH. And then the citric acid cycle, we ended up finishing what we started by taking the rest of that organic molecule and breaking it down into 4 carbon dioxides. Those carbon dioxides once again leave as waste, but what we got out of that whole process was 2 ATP, 2 FADH2, and 6 NADH2. So in a previous refresher lecture, I talked about those coenzymes, NADH and FADH2, kind of like casino chips. So what we have right now would be equivalent to 4 actual dollar bills. That represents the 4 ATP and a whole handful of casino chips. So what we need to do is we need to take those 10 NADH and 2 FADH2 and essentially cash them in for usable money, which to a cell is ATP. So that is the goal of the final step known as oxidative phosphorylation. Again, we're going to take our NADH and our FADH2, which we have a total of 12 coenzymes, and we're going to cash them in and get out usable money, ATP. Again, that's the goal of oxidative phosphorylation. Before we get into the process of oxidative phosphorylation, let's first take a look at a very important organelle that is required for oxidative phosphorylation in eukaryotic organelles, and that is the mitochondria. So here we have a picture of the mitochondria. The mitochondria is found in almost every eukaryotic cell, and it's actually a somewhat complicated organelle in that it has 2 membranes. Luckily, the membranes have obvious names. The one that is on the outside is called the outer membrane, and the one that's on the inside is called the inner membrane. How fortunate. So now we need to look at what 2 compartments result as having this double membrane organelle. The most internal compartment is called the mitochondrial matrix. And the compartment that lies between the outer membrane and the inner membrane is called the intermembrane space. Now you don't need to memorize that word because the word inter means between, and it is indeed the space between the 2 membranes. So what's critical about the mitochondria is that you have an organelle with 2 membranes, and the 2 membranes break it out into 2 compartments, an internal compartment called the matrix, and then more of an external compartment called the intermembrane space. Keep that in mind as we move forward. Okay, let's go over the overview of oxidative phosphorylation and why we get this horribly complicated phrase called oxidative phosphorylation and what it really means. Remember, the goal, we're going to cash in our casino chips, our coenzymes. What are we going to get out? ATP, or usable money. Let's first look at what happens with those coenzymes. Those coenzymes are in a reduced form. Remember, that means that it holds the electrons in that form. As part of the cashing it in, we are going to strip those electrons away. In other words, we are going to oxidize NADH. So it is the oxidation of these coenzymes that is going to be involved in the powering of ATP. When you make ATP, what you do is you attach a phosphate group onto ADP. That attaching that phosphate group on ADP is called phosphorylation. The attachment of the phosphate group and phosphorylation sound kind of similar. So to phosphorylate something means to attach a phosphate group on. So do you see the words oxidation and phosphorylation in there? And hence you get the whole phrase oxidative phosphorylation. We're going to oxidize our coenzymes to phosphorylate ADP and make ATP. So that's where the term oxidative phosphorylation comes from. Okay, as we go through the process of oxidative phosphorylation, we're actually going to start with the punchline. In other words, I'm going to have you ignore for a moment what's going on with the coenzymes. And instead, I want to turn to how do we phosphorylate ADP into ATP. And in order to understand how this works, you first have to really kind of get a good handle on what ADP is and what the phosphate group is. So I liken adenosine diphosphate, which is ADP, to putting on snow chains. If you've ever put on snow chains and you wrap the chains around the tire, you usually have to put 3 bands across in order to keep the chain in place around the tire. Now, each band represents one phosphate group. So when we have the nucleotide, what we are looking at is 2 of those bands next to each other. One band for one phosphate group, another band for another phosphate group. So if you've ever put on snow chains and you put that first band across, it's not that hard. By the time you get to pulling the second band across, it's considerably more difficult. And getting that third band across seems almost impossible, especially if you're putting them on ice. So think of getting that third phosphate group on like pulling that third band across. It's really hard. And it takes a lot of energy to bring the ADP and that phosphate group together and latch on that phosphate group. So, can it be done? Yes, but it requires a huge amount of strength or energy, just like pulling that third band across requires a huge amount of strength. So, is there energy? Sure, but it's going to require a lot of energy to really shove that last phosphate group on ADP. There is an enzyme/transporter that can do it. It is called ATP synthase, and it is so wonderful at phosphorylating or shoving that last phosphate group on ADP that every single organism on this planet uses it because it's so good. So what we're going to do is we're going to first look at how ATP synthase works. Okay, ATP synthase is actually has 2 functions in one. It is part enzyme. Remember that enzyme means to basically run a chemical reaction. The chemical reaction that we're going to run is we're going to take ADP and we're going to take that phosphate group and we're going to shove that phosphate group back on or reattach it. That's the enzyme part. The membrane transporter part means that we allow something to move across the membrane. If we take a good look at this picture here, you'll see that we see the mitochondrial parts. This being the inner mitochondrial membrane, this being one space, the intermembrane space, and this being the other space, the mitochondrial matrix. Notice that ATP synthase, this huge protein, is embedded in this membrane. And you'll notice that it allows something to come down. That something is a hydrogen ion. So here's the analogy I want to use. Imagine for a moment that I am ATP synthase, which means that technically I would have a nice little channel in my protein that would allow a hydrogen ion to come down. Now, as part of my enzyme function, I grab ADP and I grab a phosphate group, but they don't want to go together because it's too hard. I don't have the energy myself to latch them. If you're ticklish, this will make sense. I'm pretty ticklish, so I'm going to imagine that when that hydrogen ion comes down, it causes me to essentially be tickled, and I contort in a very weird fashion, almost against my will. And that is exactly what ATP synthase does. As the hydrogen ion comes down, it goes, oh, tickle, and it pulls those ADP and that phosphate group together, and they end up latching, releasing ATP. So it is the tickling, if you will, of that hydrogen ion coming down that membrane transport of ATP that actually causes the contortion of the bottom part of ATP synthase, bringing ADP and the phosphate group together, latching it and making ATP. The entire process is known as chemiosmosis. So if you read about chemiosmosis or you hear about it in class, understand it's referring to the whole idea of somehow coupling the energy of allowing those hydrogen ions to come down with the contortion that allows that phosphate group and that ADP to come together, latch, and release something that we know as usable money, ATP. So here we've got the punchline. The punchline is we know that we can somehow couple the energy in releasing a hydrogen ion down this ATP synthase to fueling or phosphorylating ADP and making ATP. So again, at this point, we're coupling what is essentially allowing hydrogen ions to come down ATP synthase and coupling that to make ATP. So at this point, your question should be, where do the hydrogen ions come from? Because clearly, in order for this to happen, we have to have a very concentrated area of hydrogen ions. And in order to make a concentration gradient of anything, that in and of itself takes work. Remember, as part of the molecular transport, molecules and atoms, and especially ions, don't like to be crowded together in the same room. So they really want out. So that's really where the energy comes from, is the fact that those hydrogen ions are crowded together up in this intermembrane space, and they want out so badly that they are willing to take any pathway, even the pathway through ATP synthase. So in order to force them to come out ATP synthase, we really need to crowd those hydrogen ions up there. So now the question is, how do we get the crowding? And for that, we need to take a closer look at what's going on in the mitochondria with the membrane, and more specifically, something that is called the electron transport chain. If you guys have some pens or colored pencils, I encourage you to get them out now, along with a blank sheet of paper, and draw what I'm going to draw over here. By drawing it out, hopefully you'll be able to see the processes that work to create this crowding of hydrogen ions. If you need to pause the video at this point, go ahead and do so until you're ready to start. Okay, so the first thing that we're going to draw is we're going to go ahead and draw part of our membrane. And in that membrane, I'm actually going to add a series of membrane transport proteins. In fact, I'm going to add 4 of them. And I'm using the purple to show this. Okay, what I have here is I have a series of 4 proteins embedded in the membrane. Let's make sure that I label everything correctly. This membrane here is going to be the inner membrane. up here, in this space here, this is going to be the intermembrane space, and down here, this is the mitochondrial matrix. Okay, these 4 proteins right here all belong to something called the electron transport chain. Think for a moment about what that phrase means, electron transport. Doesn't that mean to transport electrons? And along a chain would mean something like one to the next, to the next, to the next, etc. So, these 4 proteins basically pass the electrons around, similar to the game hot potato. So when this first guy gets the electrons, it's going to pass it to the second guy, which is going to pass it to the third guy, which is going to pass it to the fourth guy. Now, by itself, passing the electrons does nothing. But remember that electrons are going to be negatively charged, and that negative charge actually attracts a positively charged ion, such as hydrogen. So, for a moment, let's assume that we have our electrons and they're being passed. So I'm just going to use E with a negative sign, and we're going to assume they pass here, and then finally down to here. Notice 3 of these 4 membrane proteins actually span the entire membrane. Because of that, that means that they can also function in transport. In other words, you could imagine that this first protein has little hands down here that could grab something and pump it up to the top into this other space. So what would be attracted to an electron? A hydrogen ion. So as they hold the electrons, they actually have the capacity to grab hydrogen ions that are on the matrix side and basically compel them to come up into the intermembrane space side. So this electron will pump 1 hydrogen ion. Then it will get transferred to the second protein. Now, unfortunately, this second protein doesn't span the whole membrane, so it can't participate in this pumping, but it can transfer it to this one. As soon as the electron's here, another hydrogen ion is basically pulled up into this space. And same thing when we transfer it here, another hydrogen ion is pulled up into this space. So this shows you the purpose of the electron transport chain. The purpose is not necessarily to transfer electrons as it might seem, but rather to actually pump hydrogen ions up into this intermembrane space to create the gradient. And why is that gradient needed? Because when you crowd these guys together, they want out. And where do they come out? They come out via ATP synthase. Okay, now that we've drawn this, we're going to take a bigger look at the entire membrane and the electron transport chain, and we're going to see how NADH and FADH2 play a role. Okay, so here we go. Again, how is the proton gradient created? We know that we're going to need some type of an electron transport chain, as I've shown, but then the question is, where do those electrons come from? And the answer is the coenzymes. This picture here basically shows you what I've already drawn. So before we get into some of the details, let's make sure that you recognize what's on this page. You'll notice right here we have the inner mitochondrial membrane as we drew. This space down here will be the mitochondrial matrix. This space up here is the intermembrane space. And we have 1, 2, 3, 4 proteins that participate in this electron transport chain. Now, you might notice the pathway drawn by this, in this case, an orange line going up and down. That pathway is the pathway that the electrons take. That is playing the hot potato with the electrons. And anytime one of these membrane transporters gets their hands on an electron, what they do is they reach down into the matrix, pull up a hydrogen almost like a magnet and then pop it through to the other side like this. So the first guy grabs a hydrogen and pops it through. Then it passes that electron over to the next guy. The next guy is a little guy, so it doesn't actually pump anything, but it can pass it over to the third guy. The third guy, when it gets the electron, reaches down into the matrix, sucks up a hydrogen ion, and then pumps it through before finally passing it over to the fourth guy, which does the same thing. Sucks up a hydrogen ion and pumps it through. So, now that we've covered this, we need to make sure that we understand where the electrons are coming from and where they're going at the end. The electrons are coming from NADH, as shown here, and FADH2 here. Remember I said the cashing in was going to involve some sort of oxidation, or stripping those electrons away. So what happens? We take FADH2 comes up and basically dumps their electrons to this first guy. And then of course that NADH becomes oxidized. Same thing with FADH2 here, dumps the electrons and becomes FAD. So it is the oxidation of these coenzymes that drives this entire electron transport chain. As a result, we end up with the crowding or the gradient of hydrogen ions up at the top. Because they're crowded and they don't want to be next to each other, then they are compelled to go down ATP synthase, which is shown over here, and that drives the phosphorylation to make ATP. So putting it all together, what we have is we have an electron transport chain that is responsible for converting the energy in a reduced form of a coenzyme into a hydrogen ion gradient, and then we move over and we allow that hydrogen ion gradient to come down ATP synthase in a process known as chemiosmosis, and in that case we make ATP. And again, the entire thing is called oxidative phosphorylation, oxidizing the NADH, phosphorylating the ATP through this fairly complicated chain of reactions. Very last thing we need to cover is the role of oxygen. You might notice here that oxygen is what's called the final electron acceptor. In other words, when the electrons get to this fourth protein, they need a place to go or they'll get backlogged. So oxygen, which loves electrons, sits down at the bottom and says, me, 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 I'll take them, and actually strips them off that final protein. When it does that, it grabs two of those hydrogen ions and becomes water. So that is the role of oxygen. In fact, that is the only role of oxygen. If you've ever wondered why you have to breathe oxygen, that's it. Because oxygen is required to pull those electrons off and keep the flow going. As long as the flow keeps going, you keep making your gradient, you keep making ATP. The minute you get a backlog, no more ATP, death. I know it's a little gruesome, but it's the truth, which is why humans can't go more than 3 or 4 minutes without oxygen. We lose the money to make our cells run, and our cells die. So that concludes today's refresher lecture on aerobic respiration. Thank you.